Okay, good morning, everybody. It is my pleasure to introduce our guest speaker today for Grand Rounds, Dr. Dave Murray. Dr. Murray is Assistant Professor of Lab Medicine and Pathology at the Mayo Clinic in Rochester. He is a consultant in the Division of Clinical Biochemistry and Immunology and co-director of the Protein Immunology Laboratories. That's a lot of titles. And also, I think, a consultant in the mass spec core facility there. Yeah, I'm not on, okay. I don't talk maybe loud enough. So Dr. Murray received his PhD in polymer chemistry from the University of Akron, and then he went into straight as a polymer chemist and worked for 10 years as a chemist at Dow Corning and Eastman Chemical Company, where he had was able to successfully file 24 patents, and then decided that he just wanted more education, so went to medical school, and then did his residency at the Mayo Clinic in AP and CP. He has won numerous awards. He's very active in AACC, the American Society for Mass Spectrometry, CAP. He's a member of the Immunology Committee with Drs. Linden and Crossan. He has 2 grants funded by NCI currently involving biomarkers to detect early progression of monoclonal gammopathies. And he has close to 40 peer-reviewed publications, and of note, 11 of those have just been within the past year. So a very productive year. And if you kind of go to the Clinical Chemistry Journal website, you'll see actually a lot of the work that is being highlighted today. He has 2 articles in there where Dr. Murray is last author, and so it's very exciting. And so he'll be talking to us today about a new way of looking at M proteins and immunoglobulins beyond gel electrophoresis.
Thank you for the invitation. I'm really happy to be here in Minnesota. I didn't have to come too far, no airplane flight, just drive on up. And so I'm going to talk to you today about the work we've been doing in the Protein Immunology Lab. We've been trying to adopt mass spectrometry for the last probably 4.5 years. We've been working on this, and I'm just going to show you the results of that work. And I've been calling this Beyond Gel Electrophoresis. I hope to convince you that it's time to look at serum immunoglobulins again. I was telling people last night I read Dr. Nagy's book, I don't know, Modern History of Modern Immunology, and they talk about how at one time at the Basel Institute, immunoglobulins were everything until T cells came along, and we kind of forgot about immunoglobulins. So I want to bring us back to the field of looking at immunoglobulins again. All right, so I do have to disclose we did file intellectual property rights around some of this. So what I want to do today is review the structure and function of immunoglobulins so we can think about them again, so you can think about them in terms of their mass spectra, because that's where we're going to head. And then I'm going to show you how we've been using just the light chain portion of the immunoglobulins to kind of look at diseases and immune responses beyond just monoclonal gammopathies, but that's where we've done most of the work. And then I'm going to give you a bunch of cases that I've been using the mass spectrometer on to kind of help us in the lab. So that's my goal today. So these immunoglobulins are fascinating molecules. As a polymer chemist, I find these very fascinating. They have a really nice structure here. And, well, anyhow, you're going to notice a little bit of a Halloween theme. I told them I would never do this at the Mayo Clinic probably, but I'll do it here. All right, okay. So they consist of 2 heavy chains, and the heavy chain is where we get the class from, right? The G, A, M, D, and E come from the heavy chain. And then they have 2 identical light chains, except for now we know that IgG4 is not that way, right? IgG4 can swap half molecules, so those are some new learnings that are coming out of immunology. But for the purpose of this talk, we're just going to talk about 2 identical kappa and lambda light chains. And to remember, they come from plasma cells, the final stage of B cells when they finally hit the end of their differentiation. But long ago, Dr. Dury did sort of a synthesis rate. I always find this fascinating that plasma cells can outproduce liver cells. They are an amazing champion at making proteins. And so we forget that it's the second most abundant protein in serum, and so there's a lot of function, a lot of structure. There's a lot to still learn from these yet, and I think we've gotten a little bit too familiar with them. So they have this FC portion. Remember, this is the portion of the molecule that then controls the function, whether it interacts with complement or FC receptors, those kind of things. And we know that glycosylation, from the monoclonal therapeutics, they spent a lot of time on the glycosylation of the heavy chains. So we know that that's very important. We have this hinge region, which makes them susceptible to certain enzymes, and we may talk a little bit about that. But then this is really the functional end, right? This antigen binding. And this is where all the diversity comes. So the polyclonal background comes out of this variable region. And we're going to take advantage of that. And that's really the idea behind everything we're doing is this antigen binding region. And they also have disulfide bonds. And these are the ones that we're going to take advantage of to denature the molecule, to put them into heavy and light chains later in the talk. So if you remember that one of the reasons that make them so complicated is the fact that the B cells will somatically change their DNA to make a unique immunoglobulin for each plasma cell. And if you look into classical immunology, when you do the combinational statistics on this, you get these numbers that really intimidate you, like 2 × 10 to the 12th different possibilities, right? So this is sort of the challenge we have looking into the polyclonal background. But I'm going to show you data that's emerging that maybe we're not anywhere near that in terms of circulating immunoglobulins. But I think the jury's still out on that. And since we're talking about mass, I have to get you to think about the mass of these things. So just keep in mind that the light chains are somewhere around 25,000 Daltons. All right? So keep that number in mind. And the heavy chains are twice that. Okay? So just keep that in mind as we're talking about this. So what about our clinical eyes? So if you think about, okay, what are we using clinically now to look at immunoglobulins? All right. So we classically have been using nephelometry to quantitate. So you can look for the IgG, AM, D, and E, but that basically just gives you sort of like the last name, Smith, Murray, whatever, okay? You can use antigen specificity, right? You can put an antigen down and pull down the immunoglobulins. That's what we do with ELISA and for ANAs and for all the classic autoimmune markers. But we really don't know what we're pulling down a lot of times. You don't know, are you pulling down 1 clone, 3 clones, 100 clones, 1,000 clones? I'm going to show you some data on that during the talk that mass spectrometry can help us with. We can use separation. We separate these out using serum protein electrophoresis, and that's what we're going to talk a lot about today. But molecular is somewhat limited, I think. So I've been saying that immunoglobulins are a great target for proteomics because, you know, the studies that look at the entire repertoire have been done on zebrafish, and in order to get all the DNA, you've got to put the poor zebrafish into a blender and blend up the zebrafish to find all the DNA that's associated with B cells, and you really can't do that with human beings, even though it's Halloween. So at Mayo, when we think of electrophoresis, we think of Dr. Kyle. Dr. Kyle did some of the first electrophoresis. So I had him write down the history, and I put it here in this slide. This is a picture of Dr. Kyle with Dr. Waldenstrom. They were good friends. And that's Dr. Kyle now. He's still active, still in the lab, still calling me, telling me we have too many IgM mugguses in the lab, but stop calling so many of them. So he's very active. But you notice that serum protein electrophoresis started at Mayo Clinic in 1955. So we've been using protein electrophoresis since 1965. And it really wasn't, Dr. Kyle really published this first diagnostic criteria for electrophoretic patterns in multiple myeloma patients in 1960. And he said this was before Waldenstrom even proposed the fact that you'd have monoclonal and polyclonal gammopathies. He did that in 1961. I was kind of shocked to see it was that recent that we came up with the idea of monoclonal, polyclonal. But anyhow, so I put this up because this technology is pretty old. We've been running pretty much the same way since 1967 in our lab with serum protein electrophoresis. And being a trained chemist, I came in, I thought, oh, we need to get rid of these gels. They're pretty time consuming. All right, so this is it. So we're going to go to a little more individualized approach to looking at immunoglobulins. Again, we're going to take care of this variable region. And we're going to use this as a fingerprint to find the identity of the molecule. And this just kind of shows, this is a sequence we had for one of the patients, myeloma patients. So this is the variable heavy chain V3 region. We can use that as a marker, all right? And then we're going to use high resolution mass spectrometry to do that. And that scares some people, maybe more than my symbol there, but it's really not that scary, all right? Everybody tells me, do I need a PhD to interpret these things? And I'm going to convince you, hopefully I convince you that no, you don't. All right, so when we started, we thought, okay, we're going to use traditional shotgun proteomics to kind of look at these. So this is where we started. And while we were doing this, this paper came out by Decker, the group over in the Netherlands. And they kind of beat us to the punch with the concept. But what they did was they started with a monoclonal. So they're using myeloma, they started with a monoclonal drug, which they had the sequence for. So they took adalimumab, all right? They took the pure drug, all right, and they added trypsin, and they generated the peptides. Then they did LC-MS. And then they pretended like they didn't know the sequence. So they used some of the de novo software out there. Peaks is what they use. It's a program that helps you rebuild the protein structure without any database. Because remember, these variable regions are not in any of our protein databases. We don't have a good catalog of these variable regions. So you can't just go for other proteins, go to the Swiss Pro and pull out all the variable regions. They're just not all there. So they did this de novo sequencing, and then they looked at what they got from the de novo sequencing to what the actual structure was. So they knew that the de novo sequencing did pretty well at picking up these variable region peptides. And then they made this list, and then they picked one of them. Then they took the drug, spiked it back into the polyclonal background to see, okay, now that we have that peptide, can we find it? Like finding a needle in a haystack. And then instead of doing shotgun proteomics, they kind of did the targeted miRAMM approach, right? So they could really hone in on that peptide they were looking for. They added some internal standard and they quantitated. And to my surprise, as they went far, they were able to quantitate this thing far below the polyclonal background, meaning they were picking it out down into the femtomole region. And I thought, wow, that's pretty interesting, because trying to find one of these I think is like finding a needle in a haystack. And so we've used this actually at Mayo. We developed some testing on monoclonal therapeutics. It's a very simple method to follow the drug levels in some of these patients. So we went on to do this. Dave Barnage is a mass spectrometrist at Mayo who's been my partner through this whole thing. He and I have gone through a lot of these. So we just took a series of patients where we had some of the plasma cell sequence, the variable region sequence, and we went through and did the same thing and saw, okay, we can actually do this on myeloma patients. If we know the sequence, we can go in, find the peptides, and quantitate them far below the polyclonal background. So we published this work, and I have Dave here as the grand wizard of mass spectrometry today as his Halloween costume. But it was really nice work, but there's some limitations here. And there's some real limitations here. I think there's more trick here than treat, because when you start thinking about doing this clinically, you got to ask yourself, so you need to find a unique peptide for each patient to follow their myeloma. And what we found was, to get confidence that you were on the right variable region, we found that we needed an M spike. So we needed an initial sample that was above 0.8 grams per deciliter. All right? And a few of the patients we could not find unique, like 3 out of the 40 we did, we couldn't find a unique peptide, which that's a problem. And de novo peptide identification is difficult. This isn't something you can do routinely. You've got to go in, you do this thing called spider homology. You find a peptide that kind of matches close to something in the human database from the variable region, and you got to convince yourself that you're not on some addict of trypsin or some other protein before you can do this. And then we said, okay, do we need an internal standard? We need an internal standard for each patient. So patient Joe Smith is here today. Get Joe Smith's internal standard off the shelf. That didn't seem real practical. And then we started asking ourselves, well, how unique are these peptides, these variable region peptides for each patient? So again, the same group out of the Netherlands did another fascinating. I find this very fascinating. So they took pure polyclonal, they took healthy individuals, they isolated their IgG fraction out of these healthy individuals. They added this IDES enzyme. This enzyme comes from bacteria. It's one of the mechanisms bacteria use to evade the immune system. It will cleave it into the FAB-FC portion. So you can't respond to the attachment of immunoglobulins to the bacteria. Pretty clever. And then they purified just the FAB portion, all right? And then they added trypsin, and then they just did traditional shotgun proteomics, all right? So in your mind, I want you to get a number in your mind. How many peptides do you think they found for one average for each individual patient? Keeping in mind the 10 to the 12th number, those kind of things. And I was kind of shocked. This number, it's high, but it was 41,000. Well, 41,000 unique peptides. So that puts you in the category of maybe 30 to 40,000 circulating immunoglobulins in my mind. We're doing experiments to try to verify that, but not in the near 10 to the 12th range. So that I find that interesting. So then they did this for 7 healthy donors. All right. So how many percent peptides were unique to that one individual? Put a number in your mind before I show you. There's only 37 peptides out of the 41,000 that only one person had. So we share peptides in our variable regions pretty consistently among individual to individual. And so that scared me away from this technique quite a bit. And how many peptides were shared by all individuals? About 83% of them, you can find them in all 7. So we were more common in the variable region. So I thought, okay, this is probably not the way to go at this point. So we decided we needed a more practical approach to this. So we were sitting around one day and said, well, why trypsin? Let's just look at them intact. So we tried to look at them intact, but this glycosylation here does a couple things. It really kills your ionization efficiency. And these are pretty high mass. The resolution up here isn't that great. So we thought, okay, well, what if we just look at the light chain? All right. And so this is what we're going to focus on from here on, is that we're going to decouple the heavy chain from the light chain, but that light chain still has a variable region that's unique to that individual clone. So here's the basic paradigm. This is probably the most important slide I'm going to show you, because if you follow this, then you'll follow the rest of the talk, okay? So the idea is a plasma cell makes a unique immunoglobulin, all right? So if I break that into the heavy and light chains just by simple reduction, so we're adding T-sept or DTT, you can do that pretty easily. Then you can do a mass spectra on that, and so you'll get a mass for the heavy chain and a mass for the light chain. So in your mind, the mass becomes the identity of the clone, all right? So the clone's going to be identified by its unique mass, and the intensity will tell us clonal abundance. So that, if you follow that, then you'll follow the rest of the talk. So I just put this, so if you have 4 of them, you get 4 unique. You get 4 unique clones, right? But if you get 2 of the purple, now it's going to outproduce the rest of them, so you'll get a spike, just like you do in SPEP, all right? So that's kind of what we're going to be doing for the rest of the talk here. We named this thing miRAMM because I kept working with the hematologist, and they kept saying, I don't understand, top down, bottom up, what are you doing? You need to put a name on this. So this is the name we gave it, miRAMM. And I credited Angela Dispensary with this name, so she's the one who said, you've got to give it a name. So she's the wise owl who gave it the name. All right, so this is the first case. So I'm just going to show some cases now of how we've applied this. This is the very first case that I used this in. So we had a 57-year-old male came to the ER. He was fatigued. He had joint pain. He had fever. And so it kind of looked like he had pneumonia. He was febrile. He had large lymph nodes, though. He had some back pain. He was anemic. And all these things caused them to get a serum protein electrophoresis. So this came to me in the lab, and this is what it looked like. So you can see that the total protein's very highly elevated here. And, sorry, got to go back. Very elevated. The gamma region was very elevated. And so you kind of see this. You're thinking, well, maybe this is a cryo. I thought, well, maybe this is just liver failure pattern. And then we usually add reducing agent when it looks like this when we do the IFE, and this is what the IFE looked like, and people are saying, well, should we gate this or not? Okay. And so we were sitting around, I was thinking, I don't know. So before I did anything, I called the physician. He's like, I don't really think they have myeloma. So I was thinking, well, this probably isn't. So I said, okay, we're going to prove it. We're going to order the free light chain ratio and show that there's no clonality to the free light chain. But if you look, this didn't really help because this pointed to a kappa monoclonal based on the free light chain. Sorry, I keep hitting that button. So we went on to do the heavy light. So the heavy light reagents we have in our lab are unique antibodies that can differentiate IgG kappa from IgG lambda. So the antibody is specific for unique epitopes between the heavy and light chain. And when we did this, okay, we were suspecting that maybe there was an IgG monoclonal protein. But you look, the ratio was normal. They were all elevated. But the ratio was all known, but there seemed to be an IgM monoclonal in here. So we were kind of stuck. What are we going to do with this? I could have just said, can't rule out, moved on, but we decided to try the mass spectrometer on this one. And so at that time, in the state of the art, we were using a QTOF to do this. And this is what it looked like. So this was a normal patient. So you can kind of see what you'd expect. Here is a whole gamish of stuff. And we really didn't have much resolution, but we knew that if we added a monoclonal drug up to a 1 gram per deciliter M spike, you can see it. You can see all these peaks that are coming across. And this is what I said. I would never probably show this to hematologists again. As I said, what do you mean monoclonal? Because they can't get the idea that there's multiply charged light chains in here. But anyhow, so then we took that same patient that we just talked about, and we put them into the, and we saw, okay, well, we didn't see any monoclonal by the mass spectrometer. So we thought, okay, well, we're pretty confident calling this just a polyclonal background. A lot of work. Between that slide and this slide was a lot of work. So we spent a lot of time optimizing the chromatography, the conditions, till we got to this, this kind of scenario. So that same patient today would look like this. And what I'm showing here is on the top, this is the total ion chromatogram. This means all the ions that are coming out. So here you see the salts coming out from the serum, and then you get to see these are purified, by the way, for immunoglobulins. So you're seeing here, this is where the light chains come out, the heavy chains come out over here. And when we go in here and look at the TOF data, and the way we did it, we started to get this as the multiply charged spectra, which then you can deconvolute to this mass spectrum. And so we looked at that and thought, well, that's interesting. In retrospect, it should have been obvious, but at the time we were like, what is that? Why are we getting two humps? So we were asking ourselves, what are these two things? And we thought, well, it kind of looked like the kappa lambda ratio for the light chains. That's sort of what it looked like for us. So we did some work, and lo and behold, we kind of proved it to ourselves by the fact that we went in and got the sequences for lambda chains and kappa chains, plotted out the theoretical mass based on the peptide sequence. We came up with this diagram here, and this is what we see on the mass spectrometer. So we convinced ourselves that this is really the kappa lambda ratio. And so that gives us another handle that I'll show you later that helps us in looking at these diseases. So what about this patient? So this patient then, if you follow his history, he actually ended up having a really high titer of CCP and RH, so he had rheumatoid arthritis. So he had a really history of autoimmune disease. So the diagnosis here ended up being hyperviscosity syndrome, secondary to autoimmunity. He got plaques and Rituxan, did much better. But we at least avoided a bone marrow on this patient. All right, so I'm going to give you the tale of three scary cases. So these are cases that I've run across in the lab using the mass spectrometer, comparing to the gel, which I think show the power of the technique. So this is the first case, 26-year-old male. He came in from the outside, and in his history he had a 6.5 gram M spike, but the bone marrow biopsy was negative for plasma cells. But he had this long history of prostatitis that resolved, and he had a large lymph nodes. They did a biopsy. They were trying to rule out lymphoma. They weren't really sure. And when he came to Mayo, he also had a cardiac mass. So he ended up going to the heme service under Dr. Witzig, and we got his serum, and this is the protein electrophoresis that we got at the time. So again, very elevated. Outside they called it a co-migrating G kappa G lambda. Anytime I hear that now, I get a little bit suspicious, but he had a G kappa G lambda, and so he thought, okay, well, this looks polyclonal to me. This doesn't look monoclonal. So I did the free light chain ratio on this patient, and look at this free light chain ratio. So free light chain ratio is 66 to 8.2, 8.14. I'm like, oh, maybe this is monoclonal. And at that time, this is the results that went out. Dr. Witzig was still, they did another bone marrow biopsy on him because they weren't really sure. Came back negative for clonal plasma cells. But when we put him on the mass spec, this is what he looked like. All right? And what you're seeing here is something that's quite different here. If you look at this kappa to lambda ratio, and I didn't know the immune system could do this kappa lambda ratio is 5 to 1. It's very skewed from a normal person. All right? maybe explaining why this is also skewed, right? Maybe this is all polyclonal, right? So this ended up, so finally someone decided to do a subclass on this guy, and this ended up being IgG4 related disease. And so it just showed that we were kind of fooled by that kappa to lambda ratio. And I think that's one of the advantages of the mass spec. It may pull us away from using kappa and lambda ratios as the marker for clonality. And we've seen other patients like this, and in particular Sjogren's patients. A lot of the Sjogren's patients look like this, but some people think that's an IgG4 related disease anyhow, but Sjogren's patients look this way. So I'm not sure what the mechanism is that you can stimulate all your kappa producing plasma cells over your lambda cells, but it can happen. And we've also seen it reverse too, that some patients will spike lambda over kappa. We're not really sure what to do with that yet, but it's something we've never seen before, we've not been able to do. All right, here's the second case. So this is a patient, 67-year-old male, bone pain, anemia. Yeah, I had renal failure, all the classic myeloma symptoms, right? And this is the SPEP I had on this patient. And I was looking into the gamma region, and I didn't really see anything. But the reason I sent it to mass spec, I thought, well, look at that. That G lambda is a little darker than the G kappa. And I thought, well, that'll be interesting. Let's see what the mass spec shows. And lo and behold, I was shocked to see this was all monoclonal. And we did further investigations, and what we saw is the heavy chain on this particular myeloma patient had a variable glycosylation pattern. It was probably spreading it out on the gel, and we couldn't see it. Now, the patient did go on to get treatment. Probably might have been a non-secretory myeloma if I wouldn't have seen it, but still, this was actually multiple myeloma. All right, another case. So this is the case that really taught me the power of the technique. And what's interesting was my daughter was working for me for that summer, and I had her in the lab, and she brought me this on a Friday afternoon. So this was a 23-year-old male, came in from a referral. You can kind of see here the lab was questioning whether we should gate this G kappa, and by this time I was sending a lot up to the mass spec. And so when we looked at this patient, this is the one that kind of opened my eyes. This is what this patient looked like on the mass spec. So you can see an oligoclonal response, which you would never see. You can't read that into the gel. Right? So this just happened to be an HIV positive male who had a low CD4 count. So we were probably just seeing whatever he was responding to with the infection. And since then we've gone in and looked at it. Well, if you go into these hypergamma patients who are sick, you can see clones. We're not sure what to do with them yet, so we're starting to investigate that. But you can see the clonal profile actually in some of these hypergamma patients. So I think it's a fascinating study to try to figure out what these clones are and what they're responding to. This just shows a patient with ANCA vasculitis over a series. So this came out of the RAVE study that was done at Mayo. So this is a patient over a 4-year period, and we just kind of put them through the process. We don't know what to do with this, but you can see the clones come up, they come down during disease states. We're not really sure what to do with this yet. We kind of published this just recently, and hopefully we're going to get some more insight. So Dr. Specs is really interested in following some of these autoimmune patients over time and trying to figure out what these are. What I do know, they're not the PR3 MPO antibodies. We worked hard to try to prove that, and they are not that. So there's something else. So I've been saying this for a while, is that it's interesting because plasma cells are doing some sort of equivalent of a PCR for us, but it's in response to some stimuli. So I think this is another thing I've been trying to coin, is this plasma cells doing disease-specific PCR. This case just happened last week. We just finished this case last week. We had Dr. Wylam at Mayo had this patient come in who had PAP. He also happened to have a MUGUS. And usually PAP's associated with these GM-CSF autoantibodies. And so in our lab, we ran the test for that, and the titer just wouldn't stop titering. So the question was, is his MUGUS the same thing as the autoantibody? So we thought, okay, we can use mass spectrometry to prove that. So what we ended up doing on this is we took the patient's serum. We do IgG nanobody enrichment, which I'll tell you a little bit more about in a later slide. Then we reduced it. And then we put this one on an Orbitrap, and so this is the total ion chromatogram up here. This is the peak for the monoclonal. That's the heavy chain. And you can go in and interrogate this for the mass of the clone. So this happened to be a light chain MUGUS that also had some glycosylation on it. So the light chain had glycosylation on it. So that was his MUGUS clone. Then we just took the same patient. Instead of going directly to reduction, first we purified it against the antigen. So we took beads coated with GM-CSF. Then we washed off and eluted, so we had the purified antibody that was the autoantibody. We did the same experiment here, and lo and behold, you can see the masses matched up perfectly. And just, I was worried about the fact we might be getting non-specific absorption onto the bead, but you can see the reduction in the polyclonal background, that we really did purify this for the antigen itself. So we thought this was an interesting case where you can show the mass of the late chain between the Muggus clone and on the autoantibody was the same. And so they're going to go on and probably treat this patient with, try to get rid of the Muggus to get rid of the disease. So interesting application. I put this in for John Crossan because I know how much he likes celiac disease. So we've done sort of the same thing with celiac disease. You know, when you get a positive anti-TTG titer, you ask yourself, well, are all the patients the same when you get a anti-TTG? And so what we did the same thing here, where we took, negative controls. These were patients sent into the lab with suspected celiac disease that didn't have any anti-TTG antibodies. So we first purified just their IgA fraction. So these are all the clones of IgA. They're nice and smooth, and then we added this anti-TTG bead and eluted the clones off of there. And since they were negative, we shouldn't see anything, and we didn't in the normal controls. Maybe one little something stuck to the bead here. But then you start looking at these patients, and I hope this is projecting well. But you can kind of see the clones. So you can see that most of these celiac patients have some sort of response in their polyclonal background. But the clones that you see don't necessarily line up with the ones that were sticking to the beads. And so you see this patient here has one major clone dominating what's sticking to the antigen, whereas this one almost sort of has a polyclonal background. So the reason I talk about that is because titers never seem to work for a lot of these autoimmune diseases, and it's because if you're in this condition, you could be in antigen excess and not even know it. You could be in the hook effect and not even know it. So I think this is just another interesting application of that. All right. So now we're going to go on talking about miRAMM in monoclonal gammopathy. So up to now, most of the work I've shown you has been done on a high-resolution mass spec. We realized that wasn't going to work for a large screening. So we actually came up to the University of Minnesota. We rented your MALDI up here, and we came up and ran some experiments here on the MALDI math, Tom Kirk's lab. So we came up here and did this. So what we do now is the process is we take the human serum, we use these camelids, which are also called nanobodies. They're single domain. Camels have a different immunoglobulin structure than humans. It's a single chain, and they can go in and they can clone just the variable region portion of that. And so they can make these small little specific, what they call nanobodies, that you can use to pull down the human immunoglobulins. And they're used mostly in the therapeutic drug monitoring for purification of drug clones, but we've adopted them for this. So then we release the light chain, and then we put this on a MALDI mass spectrometer, which is a very fast and easy way to screen. So these are the camelids, right? So we get GAM, kappa, and lambda, and then this is the process. We've turned this is the paper that just came out in ClinCam. We've turned this mass fix. I don't like the name. I've said this before, but the hematologist told me if I don't put the word fix in there, nobody's going to know what you're talking about, and no one will order it. So we put this name fix in here, sort of. So what we do now is, so we're taking these nanobodies specific for GAM, kappa, and lambda. We're adding the patient's serum, and then we're spotting these on a plate. So we do the 5 just like you would on an IFE. Then we're doing the data acquisition, and then we overlay the spectra. All right, so in a normal patient, what you see is sort of this same sort of kappa lambda. The resolution isn't as great on the MALDI. So what you can see is, so these are all the lambda chains. So this was just the lambda. So these would be all the lambdas associated with all the heavy chains, right? Then these would be the lambdas associated with IgG. These are the ones associated with IgA, and this one IgM. And then these are all the kappa chains. So by doing this, we can look at the profile in the background. Compare that to a myeloma patient or a Muggus patient. What you see now is you don't see all you see is 2 peaks. You see one that's associated with the pink, which is the kappa, one with the dark blue, which is a G. So this is a G kappa Muggus. So this is sort of how we're reading these now, and we're developing software to kind of automate this. And all this work was really done, I give credit to John Mills, and I gave him a costume today. I asked his permission. So I'm calling him Boy Wonder. So he gets Boy Wonder. And Mindy Cohen's a tech in our lab, and I call her the Princess of Maldi now. So, but they're working really hard. We're in the process of implementing this into our lab. But you get really information-rich spectra, even off the Maldi. All right. So what I'm showing here is the +1 and +2 of the kappa light chain. So you can see where the kappa light chain is. This is the lambda light chains. But you can see heavy chains on here. So you can see they don't ionize as well, and they aren't as easy to read. But that's the IgG +2, 3, and 4, 5. And you can see IgA. Rarely do we see IgM because the background quantity of IgM is usually not that high in normal patients. But this is the beauty of the nanobody. So the nanobody, if it happens to come off the bead, which it sometimes does, it's in a different mass range than humans. So you can, it doesn't interfere with the test. So that's why we like these nanobodies for this test. All right. So here's an example of the difference of a Maldi versus the electrospray. All right. So this happens to be a heavy chain disease patient. It was just a good example. So this is a truncated heavy chain with different glycosylation groups on it. So this is what it looks on the low resolution Maldi. This is the same patient on the ESI-QTOF. So you can see we get a lot of resolution, but I drive the mass spectrometrist crazy. I tell them I'm a pathologist, and I can visualize those going down to the bottom perfectly fine, because this takes me about 5 seconds to collect. This takes me 25 minutes. This is off a $125,000 instrument. This is off a $650,000 instrument. So I'm perfectly happy visualizing them going down to baseline. So, but the Maldi's really is rapid, and I know, I'm sure the mass spectrometrists in the room are probably cringing at that statement. So here is our setup. So we are in the process of validating this. So we're doing all the purification, upfront purification on a Hamilton, spotting it with this lovely little thing called the Mosquito. Pretty clever instrument that does all the Maldi plates, and then it goes over to the Maldi mass spectrometer. So this is how we're setting up. And I have one development tech, Mindy herself. She can run 400 or 500 of these a day by herself. So it's a pretty fast technique. So this is it running. So I'll show you it running in real time. Let's see if I can get this video to work. It's going to go upside down on you, because it always does on me. Maybe it's the Halloween. But okay, so already, sorry that it's upside down. The video keeps flipping on me. But you can see a spike here. So you can see these are the, this is the +2, +1 region. So you can see the spike here already in the first few laser shots. You can already see that this patient has a monoclonal protein. And so we collect a total of about 700 spots per patient right now. Going to the next patient. The video behaves. Okay, so here comes the spot, and already I can tell you this is a normal kappa lambda ratio, right? Even though it's upside down, normal kappa lambda ratio. So the video is a little jerky here, but you can kind of see how this is a really rapid and fast technique to do this. So it goes to the next spot. I don't know why the video is so jerky, but it is. But I think you get the point here, is that compared to a couple hours of electrophoresis, we're collecting these in almost real time. So we went off to try to validate this method. You can look here. So what we saw is, so these are patients that were positive by SPEP, so meaning they have above 1 gram per deciliter, right? We didn't miss any of those patients. And then when we looked at the ones that were below, that we call IFE only positive, so you don't really see the abnormality on the SPEP. So we really stressed the system out the best we could, and we had pretty good match. 2 cases flip-flop back and forth between the IFE and the Maldi. So very good agreement. And now, you know, where IFE you can't quantitate off of, and you have to use SPEP, we can quantitate off the Maldi, and the way we're doing that at the moment is to, you just take that spike. So this would be the total IgG. You have a spike here, which is the monoclonal, and then we can just integrate the area under that spike. And then we just take that area over the total area times the nephelometric value of IgG, and we get very good agreement. In fact, we can go way below the 1 gram per deciliter because we're excluding a lot of the polyclonal background here. So we've gone down to 0.02 grams per deciliter with linearity. So that's nice. So you get some new information. I find this interesting. So this is just normal patients. where we take the light chain distribution, that's 40 of them stacked over top each other, and you look at the light chain mass distribution, and then all of a sudden you see something interesting, because when we start looking at Waldenstrom's patients, we were like, wow, all the Waldenstrom's patients seem to have these heavy kappa chains. We call them heavy kappa chains. But if you look at normal IgM, the circulating IgM, you see this fraction out here. And so you can go back and figure out, well, what are these heavy kappa chains? Well, they're all kappa V2, V4, because they have extra amino acids in it. If you look at the sequence, for some reason IgM are picking kappa V2, V4, and I'm not exactly sure why, but the circulating for sure has more of that fraction. So that's new information. I'm not sure what to do with it yet. But just like the heavy light assay, we showed that you can actually calculate the G kappa G lambda ratio just by looking at the area under these two curves, and they match very well with what we did with our heavy light assay on the nephelometer. So that information comes along almost for free based on the mass. And then we started, Angela Dispenseri and I have been looking at different diagnosis of mugguses and just looking at patterns. We kind of did a little fishing here. And we had a fellow from Dr. Merlini's lab, Paulo Milani, was over visiting us. And so I like, okay, we're just going to do some fishing exercises. So we took all these monoclonal gammopathies with different diagnoses and we started looking at them. And this is what I call the standard myeloma pattern that we see off the mass spec. And what I'm showing here is this. So this is the normal. So this is your normal kappa lambda ratio for a G. So this is a patient with myeloma. You can see a spike here from the G pull down in the lambda. So this is a G lambda patient. The masses match. And the nice thing is when you go into the urine, you see the same spike and the masses all line up. So you're really confident. So this is the standard myeloma patient. But then we started noticing there's patients that are doing different things than just getting the standard pattern. And this happens to be one of them. So this happens to be a patient with renal disease. So they have a muggus, so what we call monoclonal gammopathy of renal significance. And what we started noticing is some of these, so if you look at the kappa, we don't really see much in here. This is a normal kappa spectrum. But when we looked at the urine, you start seeing fragments in here. And we've proven and convinced ourselves that these are actually fragmented light chains in the urine. So it seems like maybe the kidney's processing some of these light chains and changing them somehow. So that we think that that's useful information. Here's another very set of interesting. So we have a lot of patients with amyloidosis down at Mayo. This happens to be one of these patients. They have a G lambda. And so you can kind of see a couple things here. That this patient really isn't monoclonal, what I'm showing here. So if you look in the G bead, you can see a peak here and a peak here. And that same peak shows up in the lambda bead. So that means that this lambda is attached to a G. But if you look out in this region, what you're seeing is this big peak. It took us a while to start to appreciate these because we didn't know what they were. But you can see, so this isn't showing up in the G bead, so this must be only associated with lambda itself. So it's a lambda-free light chain. And if you go into the urine, you can see that same pattern over here. And these turn out to be glycosylated light chains. So we are seeing that amyloid patients have a lot of glycosylated light chains, and we're not exactly sure what the significance of that is, but this is sort of new information. All right, so we're also looking for a more sensitive MRD detection. That's another area that we're looking at. And now instead of using the MALDI, we're going back to that high-resolution instrument. But the nice thing is you can take the same prep, run it on the MALDI, and then take it right over to the high-resolution if you need to, and look at it for minimal residual disease in myeloma. This just shows some spiking experiments we did early with the miRAMM method. So this is a patient that had a 3 gram per deciliter A kappa. You can see the mass here is 23,334. Then we took that patient, started to dilute him into normal human serum to the point where we diluted it 1 to 1,000. By this time the A looks totally normal on IFE, but if you look at it, you can still clearly see the peak in the polyclonal background. So we estimate that depending on where in the mass spectra you are, you're somewhere around 100 times more sensitive than IFE by this high resolution mass method. So how does that work for myeloma? Well, okay, so this would be prior to treatment. So you get this mass here. You can see it's monoclonal. We usually see a subclone in most patients, small subclones in most patients. Then post-stem cell, so what you see is after stem cell treatment, they don't really go back to that nice smooth kappa lambda polyclonal background. They're kind of spiking, and you see that on gels all the time, that sort of banding pattern. But now, because we have this mass resolution, we can actually pick out which one's associated with the malignant clone because the masses match exactly. So we estimate our resolutions plus or minus 1 Dalton at this point on this method. So you have pretty good confidence that you're on the right clone. But here the intensity was 580,000, here 7,000. So I'm showing the difference before and after treatment. This just shows, thanks to Dr. Kyle's database, we went back and created some results for patients over the time. So this is a patient that had myeloma that was diagnosed in 2005. They had a 4.8 gram per deciliter M spike, positive IFE, positive free light chain ratio. They underwent treatment. In 2007, they went into what we would call strict complete remission because the flow was also negative and bone marrow was negative. So everything was negative here. They relapsed in 2008, got treated again, went back to remission again in 2011. So we reran all these on the mass spectrometer, and we could detect monoclonal immunoglobulin in the whole entire time. This is the mass. So you can see this is the mass of the clone, so plus or minus 1 Dalton over that whole entire time, which is nice to know. But if you look at the intensity, so we started out at 3 million in the peak area, went down to 9,000, kind of showed it was coming back, and then it went down to 2,000 here during the second one. So we feel pretty confident this can follow pretty easily over time without bone marrow is the key here. This just shows how we envision this moving forward for MRD. This just shows another patient again. This is an IgA, M protein, pre-treatment. Day 100, you can still see it. So, but this patient at Mayo was under strict complete remission, meaning we did the flow. We didn't have any of the sequencing data yet, but on flow negative. And then at day 300, you can see it's totally gone. So this is a way to follow the patient over time to see if they're continuing to respond or not. All right, so that brings me to the last one, interesting future possibilities. So the other thing I haven't touched on yet today is the ability to kind of do top-down sequencing on these. So earlier when we did this, we can isolate in this spectra on the top. This is the monoclonal protein. You can tell the mass spectrometer to isolate that and fragment it. And so we've been doing that. And so when you fragment it, we can definitely see the kappa constant region in here. And so we can tell the difference between kappa and lambda, but in here is all the variable region information also. So we're hoping that maybe in the future we can take advantage of that because our amyloid group has been showing that there are certain sequences of light chains that are have a more propensity to form amyloid than others. So, and Angela Dispenseri is very interested in POEMS because POEMS selects a very specific variable region. So we've been working, Dave Orange and I have been working with the Center for High Mass Spectrometry down in Florida, Alan Marshall's group. And so this is not clinically practical. This is 21 Tesla magnet. This is a very expensive instrument, but it shows what's possible, okay? So what you're seeing here is, I challenge them, say, how low can you go and give me sequence? How low on an M spike can you go and give me sequence information? So what they showed was, so if you take pure adalimumab and do it, and you fragment it, they can get about, in this case, they can get 90% coverage of the sequence, which is enough to pick the gene class and the family. We don't get the exact sequence, but you get enough to get that. Here's one we took down to about a 0.2 gram M spike, and they isolated that and fragmented that, and we still had about 57% sequence coverage. So we're hoping we can use that, especially in these patients that are sick and flying cone, to get an idea of what plasma cells are being stimulated when people get sick. So this is way down the road, and it'll be a long time before we get there. All right, so I'm going to close. I've given you sort of my Halloween version, but I want to end on a positive note. So this is something I put together. It's a late Friday afternoon, but I'm just going to show it. So this is what I'm going to show you is just a patient I actually got from Janssen. So I have their permission to give this. This is someone from the daratumumab trial, and it's a little tale. I call Hello Dara, Goodbye Clone. So what you're going to see is I'm going to give you serial measurements off the mass spectrometry of a patient on daratumumab, which is the new anti-CD38 drug that's making really great strides into myeloma. So this is, remember, this is the normal patient, right? The normal kappa lambda ratio. So this is the healthy immune system. And I said all was happy in the neighborhood until Mr. Malignant plasma cell moves into the neighborhood and he decides to take over. So this is another one of these patients has a glycosylated light chain, but you can kind of see what's happening here. So this is the myeloma, all right? You can see that all the normal polyclonal background's been suppressed by the malignant plasma cell. So I said all the normal kappa and lambdas are relocated from the neighborhood. But a few weeks later, a challenger arises. So I think this is one of the reasons that mass spectrometry will also make it into this field, is that all these monoclonal therapeutics are just multiplying over and over again in this field. But because we know the mass of them, we know where they should show up. So you can see there's a spike here that's coming in, and this is daratumumab coming into the picture. All right, so we can see daratumumab coming in. He says, my territory's shrinking. Oh, what's this new clone I spy in the neighborhood? Are you friend or foe? And out comes our superhero. And this is then 3 weeks later. You can see what happened to the M spike. This is now 6 weeks later. You can see how it's returning back. Daratumumab's doing some amazing things to the immune system. And eventually you can see that it's going back totally to, you can still see a little bit of the daratumumab in here in the patient, but you can kind of see the normalization back of the immune system. So I said, who was that masked person? It's the myeloma group at Mayo. So this is the myeloma group at Mayo, and I give them a lot of credit for supporting me in this endeavor, because they could easily say no. There's a lot of change that has to happen for them to adopt this method, and I've been really thankful that they've given me support during this whole process. There's a whole bunch of other people I need to thank also. John Mills has put a really yeoman effort into this, helping me. He's the one who came up with the idea of using camelids, and so he's really pushed that for us in the lab. Mindy has been setting this up in our lab and working really hard. I mean, lots of people to thank. So I think with that, I'll close and see if you have any questions. Yeah, John.
David, that was a fascinating presentation. Thank you. Yeah. One of the theories about myeloma is that a single plasma cell reacts to a single epitope, and then all of a sudden it becomes Mr. Malignant plasma cell. Can you use the Fab peptides that you have made to identify that single epitope? And do you think it's going to be a single epitope, or do you think there'll be a number of epitopes that Fab peptide reacts to?
All that would be possible. We haven't even, I haven't even thought of that yet. So that would be a fascinating study to do. Vincent has been pushing me to take the Olmsted County database that we have and take the people who were negative when they came in and who ended up getting a muggus during the time that they were in our cohort, and going back and seeing how, if we have a more sensitive technique, how did that clone arise, and did it come out of some oligoclonal response or not? He's been pushing me to do that for a while, but I haven't had time to free up time to do that yet. But
yeah, it's going to be a single epitope or several epitopes?
I think it's going to be several, but I don't know.
Yeah, that's what your early slides would indicate.
Yeah. Yeah.
Yeah. So I liked how you showed that, like, if you went back and looked at the patients in stringing CR, you could find their colon by this method because it's more sensitive. I'm curious this is going to gain a lot of traction as a reference lab method because not everyone can set it up as easily as you can, but it takes some time. So if a bunch of labs start sending you myeloma patient samples for patients that are in SCR and you don't have any prior data, how well does it work when you don't know anything about where their spike should fall? Like, what is your lowest level of sensitivity, do you think, in terms of concentration?
Okay, so I would say for the MALDI, it's a little better than IFE, but not. So it's still in that milligram per deciliter kind of area. It's a little tricky because you can play games with the mass spectrometer, and I don't know if they're real games or not. So you can go in and isolate yourself, spread out the chromatography, narrow in on a window, and you can find clones, but I don't know if that's a legitimate way to actually find them yet or not. I think it's Dave Barnage and I have this argument all the time. I'm like, I don't know if we can get away with this, if we're artificially increasing the sensitivity by doing that or not. But if you do that, you can go pretty low. You can go down to point on that high resolution. They went down to some ridiculously low number, like 0.1 milligram per deciliter, which then you're starting to wonder, what am I really looking at? I mean, have I just now found the polyclonal background and I can find everybody this way? So it's some of the questions that we have that I think is absolutely fascinating. Yeah.
In the patient that you showed the single kappa chain over 7 years and the relapse and whatever, you were positive all the way through. So the question I'm sure people ask is, when do you start therapy? If you know that, when do you or stop therapy, given that?
So that's the next step. We've been talking about we need to show where the clinical significance is in terms of an increase. So right now the Black Swan trial, Mayo is part of the Black Swan trial, which is the push for cure in myeloma. Some people think we can cure myeloma and maybe we should push to 0 MRD. I can tell you that the Mayo hematologists are not there yet. They don't believe in pushing people to 0 MRD. So they won't treat until symptoms come back or something's increasing. So those are questions that are still out there. But I'm telling you, with daratumumab and these other new drugs, I think the game's changing, especially because these aren't seen to be toxic. They can't find LD50s for some of these drugs. So.
You could shift back to normal.
Yeah. I think Dara is going to end up like rituximab eventually. I think they're going to find all kinds of really good things to use it for. Yeah, sorry.
I have a few questions. So B cells have immunoglobulins on the surface, and we look at them to tell lymphoma or not. Have you been able to use similar method on B cells?
Yeah, we
solution?
Dave Barnage, that's how he started out. He wanted to look at immunoglobulin rearrangement for CLL, so to speak, see if we could show whether it's you can do it. It's a lot more work because you got it. The pre-analytical is a little more challenging because you got to lyse cells, recover it. It's not like the beauty of plasma cells is they're just spitting out so much protein that it's easy. But we've done some of that. I wouldn't say it's a robust method. Not sure it's going to be any better than using molecular at this point. Maybe you don't have to pay royalties to Roche or something, but anyhow. So, yeah, we've done it. I can't say that it's something I think is going to be useful. I think it'd be hard to. We did it with the plasma cells in the bone marrow, and I think the flow people are like, no, we're not going to do that. We're going to continue to do flow. Yeah, sure.
Question regarding your cartoon at the end. So daratumumab should also eliminate normal plasma cells. I'm surprised you didn't see normal pattern come back. Why is that?
The biology of daratumumab is fascinating to me. It seems to eliminate some and stimulate others, and I'm not sure why. I've asked the drug company, they just remain silent when I ask them that question. I think they know why, but I don't think they're telling anybody. Huh?
Probably working on related drugs.
Yeah, they probably are.
Maybe the patient was on IgG or something?
That's a good question. I don't think I have an answer for you. It's a great question.
Yeah, great talk. The fragmented light chains you showed in urine, is that the equivalent of the urinary light chain ladder pattern that was Harry Harrison described years ago?
Yeah,
we
I haven't.
used to see it, now we don't see it because we shrunk the gel.
It could be. I've never seen the larger gels either. We don't even use high resolution gels at Mayo because I asked one time why we're not. Dr. Kassman told me, Dr. Kyle said we find enough mugguses, we don't need to find any more. So I've never seen that. I've never seen that pattern in urine, but I see it on the mass spectrometer, so it could be. But I've never personally experienced the higher resolution gels. So.
Excellent presentation. But did you try to use this on another type of fluid like CSF, for example, in multiple sclerosis?
So yeah, we've published a paper on oligoclonal banding using this method. It's a very nice method because you can go right into the serum and compare the CSF, and so we have that publication's out there of using this for oligoclonal banding. And it's very fascinating that a lot of the MS patients select kappa. They are selecting kappa over lambda, and a lot of them are selecting the that kappa V2 V4 that I was talking about, and I don't know why. And Moses Rodriguez, I don't know if you're familiar with his work down at Mayo, but he's got that monoclonal therapeutic for MS. He came into our lab and took all Dr. Kyle's database, went through all of our monoclonal proteins and found 2 of them that actually heal plaques in MS. And they were both IgM, and they were both IgM that had these heavier chains on them, so I find that fascinating. I don't know if there's really a correlation there or not, but I find that fascinating. But yeah, so that's why I find immunoglobulins fascinating, is that there's a lot of people out there now pushing the fact that immunoglobulins aren't there just to remove things. They're also there to heal and do some other things. And so, yeah, I think it's fascinating. So we have done it in CSF. Michael?
Have you found a way to code and bill for these similar to like protein electrophoresis or IDEX?
I'm sorry, what was that again?
Coding and billing? How would you code and bill for this test?
Well, you know, you're on the DIRC. I tried to get a CPT code for this, right? Made it through the DIRC, got to the CAP Finance Committee, and they shot it down, right? Because there's not enough people using this. So I've been jokingly saying this is gas phase electrophoresis, but nobody will buy that at Mayo. They won't let me use the electrophoresis CPT code, so we tried to get one. And they told me in the CAP Financial Committee that you don't want one now because you get what they call a second class. I don't know what they mean by that. A second class CPT code that once you're there, you're stuck. And most insurance won't pay for those type of CPT codes. So it's a conundrum. We're going to lose money on this assay when we bring it forward.
Could you like as a clinical pathology consult or?
Yeah. Well, I work for Mayo Clinic, and it's very conservative down there. We're not going to do anything that risks getting us in trouble. So we're going to do the general MALDI code, which is not.
That is reference lab testing, right? Because even if other people in the myeloma community want it and they send their test to Mayo, you go with third party.
So the solution is to get it FDA cleared and to get it into the real world, which is going to take a lot of time. All right.
Thank you.